Hello students, I am Dr. Rajeshwari Sinha and today in this lecture I am going to talk to you about the importance of biostatistics in scientific research. Data scientists are touted as the most highly paid professionals today who are earning even more than a chartered accountant or an engineer. The data scientist's job draws heavily on statistics. In life sciences, the huge data sets that are rapidly being generated from genome and proteome projects require to be analyzed and meaningful inferences drawn from them. In such situations, we apply various statistical tools and this constitutes the realm of biostatistics. In this course, we will discuss the structure of various statistical tools used for data validation and analysis and illustrate their application using case studies from life science studies. I hope you have an enjoyable time learning these tools and at the end of the course are able to apply them with ease on real-time data. In today's lecture, we will firstly try to understand the importance of biostatistics in data analysis, followed by an appreciation of the various prerequisites of a statistical experimental design. Once the data analysis is done, what is the typical output and what should be the compositions of a statistical analysis report. Lastly, by means of a case study, we will look at an application of biostatistics in a clinical trial experiment. This slide shows the logical flow of lecture as it unfolds. Before you start any new branch of study, there is an appreciation of its importance in your mind, isn't it? What benefits will the study give me in terms of increasing the data robustness, acceptance of my hypothesis and empower me to make prediction about the results in a very rational and unbiased manner. Having known the benefits I am going to reap from the study, I have also to equip myself technically to be able to use the various tools to my maximum benefit. Since a job well begun is already half done, we should know how to initiate a biostat-enabled study. You cannot randomly decide in the middle of an experiment to do a statistical analysis of your data, although in a very limited sphere this is also practiced. The entire experimental design has to be biostat ready right from the start. So we will learn how to initiate such an experiment. Lastly, since knowledge without application is like a book never read, we will also see how to use a biostatistics tool in analyzing a real-world clinical data set. Biostatistics has wide application in the field of public health, pharmacology, clinical trials, medicine, agriculture, epidemiology, biology, genetics, environmental science and nutrition. What is biostatistics? The branch of statistics that applies principles of statistics to the field of biology come under the ambit of biostatistics. It covers a wide area and includes designing and monitoring of biological experiments, collecting and verifying data from these experiments, summarizing and displaying that data, and analyzing the data to draw meaningful conclusions from it. As implied, these are in fact the steps in a scientific methodology. Scientific methodology. Gone are the days when we based our decisions on hearsay. On the one hand, increasingly huge volumes of data are being increasingly generated and easily generated thanks to the advanced instrumentation and computational abilities across all fields of research and particularly in the field of biomedical research. Genome and proteome projects have led to the generation of data of overwhelming proportion almost overnight. And yet, publication of research based solely on large data sets has only got tougher. This is because the sheer amount of data that exists necessitates the availability of robust abstraction tools to take the study to a meaningful conclusion. Numbers make sense only when they are analyzed in context of the variables that they represent. In scientific research, as much as in any other field where huge data sets are to be analyzed, statistics plays a major role in deciding the outcome of a study and also in gleaning meaningful information from such a study. To produce objective knowledge is difficult since our intuition has a tendency to see patterns where there is only random noise and to see causal relationships where there are none. When looking for evidence, we also have a tendency as a species to overvalue information that confirms our hypothesis, and we seek out such confirmatory information. When we encounter new evidence, the quality of it is often assessed against the background of our working assumption or prior belief, leading to bias in interpretation and scientific disputes. To overcome these human shortcomings, the so-called scientific method evolved. This is a method which helps us obtain and assess knowledge from data in an objective way. The scientific method seeks to explain nature in a reproducible way and to use these explanations to make useful predictions. To gain acceptance for one's conclusion, it is critical that all the details of the research are made available for others to judge their validity, so-called peer review. Not only the results, but also the experimental setup and the data that drive the experimenter to his conclusions. If such details are not provided, others cannot judge to what extent they would agree with the conclusions, and it is not possible to independently repeat this experiment. As the physicist Richard Feynman wrote in a famous essay condemning what he called cargo cult science, if you are doing an experiment, you should report everything that you think might make it invalid. Not only what you think is right about it, other causes that could possibly explain your results, and things you thought of that you would have eliminated by some other experiment and how they worked, to make sure that the other fellow can tell if they have been eliminated. Before a scientific study can be transformed into a body of knowledge, it goes through several steps. These steps are shown in the figure. The first step involves data collection, where all the pertinent data related to the study are acquired and the variables are defined. Based on the data, a researcher makes a prediction or proposes a hypothesis about a phenomenon. Then is the stage of verification of the hypothesis. In this stage, again data is generated to validate the hypothesis. The data may either refute the hypothesis or validate it, in which case it may have to be revised. In each of the steps outlined above, statistics plays a major role. It provides crucial guidance in determining reliable information and meaningful predictions. At the stage of data capture, statistics helps determine the sampling size as well as data collection methods. Once the data is gathered, it allows for processing of data and decide on the strengths and limitations of the results. Thus, a scientific mystery is unraveled in a very objective manner, free from bias and subject to the given set of conditions. Statistics. The online dictionary www.dictionary.com defines statistics as the science that deals with the collection, classification, analysis and interpretation of numerical facts or data, and that by use of mathematical theories of probability imposes order and regularity on aggregates of more or less disparate elements. The Merriam-Webster's Collegiate Dictionary definition is a branch of mathematics dealing with the collection, analysis, interpretation and presentation of masses of numerical data. The steps of statistical analysis involve collecting information, evaluating it and drawing conclusions. A statistician determines what information is reliable and which predictions can be trusted, search for clues to the solution of a scientific mystery without being misled by false impressions. Among the other roles of a statistician include determination of suitable sampling and data collection methods, monitoring the execution of a study and the processing of data, and also advise on the strengths and limitations of the results. It is crucial to understand the nature of uncertainties and be able to draw conclusions in the context of particular statistical applications. In a statistical study, we try to establish relationships between the population variables. However, it is imperative to agree on certain aspects of the study so that it reflects the true picture. Hence, we can draw up certain basic requirements for a statistical study which are outlined in the figure and discussed below. Proper designing of the study. After identification of the population of study, one should go for unbiased survey, keeping in mind the proper representation of the sample from the population. An observational study is based on our observation of phenomena, events around us. When we try to establish a cause-effect relationship, a randomized experiment model is used. For example, to know whether caffeine causes an increase in heart rate, we need to ascertain that increased heart rate would only be due to caffeine consumption, since the two groups would be chosen so that nothing else differed between them. Exploratory data analysis and pilot studies can also be done. Choose the sample carefully. The individual cases chosen for study should be representative of the original population of which it is a subset. For instance, in order to study the correlation of color blindness with automobile accidents, the representative sample should include automobile drivers, persons with known affliction of color blindness, as well as survivors of automobile accidents. However, young school children as well as convalescing old people may not constitute representative sample. Define a judicious sample size. For the above cited case study, can just one teenager and one elderly person suffice to arrive at a meaningful conclusion, or 20 or 50 can be considered sufficient sample size? These queries are to be well defined for selection of an ideal sample size. One criterion to decide the sample size is the extent of variation within each group. It is also important to select a suitable study type. For instance, does the study warrant a large-scale census or a small-scale cohort study? This has to be clearly stated before suggesting the possible ramifications of the study to a larger population. Biostatistics is the branch of statistics responsible for the proper interpretation of scientific data generated in the biology, public health and allied health science domains. In these sciences, subjects such as patients, mice, cells, etc. exhibit considerable variation in their response to stimuli. The most likely sources of this variation are different treatments or chance, measurement errors or other characteristics of the individual subjects. Biostatistics particularly seeks to disentangle these different sources of variation and distinguish between correlation and causation in order to make valid inferences from known samples about the populations from which they were drawn. For instance, do the results of treating patients with two therapies justify the conclusion that one treatment is better than the other? Biostatisticians regularly engage in interdisciplinary collaborations and advance their discipline through pursuit of methodological development of statistical methods for biomedical applications. Biostatistics is a broad discipline encompassing the application of statistical theory to real-world problems, the practice of designing and conducting biomedical experiments and clinical trials, the study of related computational algorithms and display of data, and the development of mathematical statistical theory. Biostatistics is integral to the advance of knowledge in biology, health policy, clinical medicine, public health policy, health economics, proteomics, genomics and other disciplines. Components of a statistical report. It is important that the results of a statistical analysis are prescribed in a lucid and comprehensive format. A standard statistical report needs to have 7 key components. These are as follows. Funding source. It is important to declare the funding source so that the reader detects if the study was funded by an organization that would be biased to a particular outcome. Blind studies. Participants who are known to the researcher and her field of study may give answers that are biased. Therefore, it would be ideal that all participants and the researchers are not known to each other beforehand. Thus, a double-blind study ensures greater level of neutrality. Selection of sample. So that we can know if the sample is representative of the entire population. Also, if the study claims to be a randomized experiment, was the sampling method really random? We will learn more about random sampling later. Measurements. We want to precisely define what we want to measure, why we want to measure it and how to measure it. Experimental conditions. This can help us see if there was any bias in the measurement process. differences in groups being compared. This is the key in the difference between observational studies and randomized experiments. In observational studies, such as the ones mentioned above, because of confounding factors in the observational studies, we cannot imply causation. In randomized experiments, we hope to be able to control for these confounding factors, and thus we can imply causation if the study is done correctly. For example, with the teen and elderly drivers, other such factors would be how often or how many miles each group drives, extent or size of any claimed effect or differences. This implies whether the results are of practical importance or not. Case study. Many researchers have run into problems while analyzing their data because of the decisions they made or failed to make while designing and executing their study. Many of these early errors arise from not understanding or appreciating the different kinds of data that their study can generate. It is therefore important to recognize the kinds of data you encounter in a biological research: numerical, categorical, and date and time oriented data, and how to collect and validate your data. Besides, it is also important to see how to summarize each type of data and display it graphically. Let us understand the application of statistical tools for the analysis of real-time data using this case study. Here, we describe selected aspects of the study pertinent to the lesson. For further details, the reader is referred to the original research article available at PubMed. The Mediterranean dietary pattern had long been thought to reduce the risk of cancer in addition to being cardioprotective. The present case study is based on a randomized trial done to prove this belief. In a study, over 600 survivors of a first heart attack were chosen for a randomized trial 2 months after their stay in a cardiac care unit. They were assigned into either a control group or an experimental group. Patients in the control group were given a diet plan close to the Step 1 prudent diet of the American Heart Association, whereas patients in the experimental group were instructed to follow a Mediterranean type diet with more bread and cereals, more fresh fruit and vegetables, more legumes, more fish, fewer delicatessen foods, less meat, and no butter and cream, which were to be replaced with an experimental canola oil based margarine rich in oleic and alpha linoleic acids. The oils recommended for salad and food preparation were canola and olive oil exclusively. Moderate red wine consumption was allowed at meals. Patients in the control group followed Step 1 diet of the American Heart Association, characterized as 30% of the total energy as fats, 10% saturated, 10% monosaturated, and 10% polyunsaturated, and cholesterol intake lower than 300 mg per day. This randomized trial suggested that patients following a cardioprotective Mediterranean diet had a prolonged survival and may also be protected against cancer. In order to accept this claim, this research group included various statistical measures to increase the validity of their data. These include univariate analysis, calculation of 95% confidence interval, as well as the two-sided p-value. In this lecture, we learnt about the importance of biostatistics in data analysis, the prerequisites of a statistical experimental design, components of a statistical analysis report, and application of biostatistics in a clinical trial experiment. As I mentioned, data analysis is a multi-billion-dollar venture in the industry now. Companies like Google, Amazon, Netflix, and Uber have dedicated data science groups. In the pharma and life sciences sector, companies are still skeptical to invest heavily in data analysis, but research groups are already generating huge data sets and validating them using data analysis algorithms. But by applying data analysis in medicine and life science research, in future many things may be possible, such as predictive modeling of biological processes and drugs, which could help identify new potential candidate molecules with a high probability of being successfully developed into drugs that can act on biological targets safely and effectively. It can help in identification of patients to enroll in clinical trials based on, for instance, social media rather than doctors' visits. Furthermore, the criteria for including patients in a trial could take significantly more factors, for instance genetic information, into account to target specific populations, thereby enabling trials that are smaller, shorter, less expensive and more powerful. Trials can be monitored in real time to rapidly identify safety or operational signals requiring action to avoid significant and potentially costly issues such as adverse events and unnecessary delays. The data are captured electronically and there is ease of data access across all stakeholders, namely research groups, development groups, physicians and contract research organizations. This easy flow is essential for powering the real-time and predictive analytics that generate business value. Thank you for watching this lecture.